Welcome to Zoology class. In the last class, we have seen by the experiment conducted by Alfred Hershey and Martha Chase. It was beyond doubt ascertained or made sure that DNA is the genetic material. And further studies have revealed that RNA could also act as a genetic material in some organisms like viruses. Anyway, the doubt that whether a protein could be the genetic material has been completely eradicated by the experiment conducted by Hershey and Martha Chase. So it was established that DNA is the genetic material. And now let us see the molecular structure of a DNA molecule. We had already studied the structure of the DNA molecule in the first year. And let us recall what we had studied in the first year. DNA is a polymer. As every polymer, as in the case of any polymer, DNA is also made up of its monomers or units called nucleotides. Unlike other polymers, DNA has two strands connected together. It has two polynucleotide chains. It is made up of two polynucleotide chains bonded together to get a ladder-like structure. And each polynucleotide chain is made up of nucleotides, several nucleotides, and each nucleotide has three components. Each nucleotide is made up of three components. They are a sugar, pentose sugar, nitrogen base, and a phosphate group. Let us see each one by one. So one of the components of the nucleotide is pentose sugar. Pentose sugar is a 5-carbon compound in which 4 carbon atoms form a ring-like structure and the 5th carbon atom is attached to the ring that stands outside the ring. And the carbon atoms are numbered from 1 to 5, as you can see in the diagram here. It is a deoxyribose sugar. The sugar is deoxyribose sugar. Because in this sugar, one oxygen atom is less than it is in the ribose sugar. Okay, it's a 5-carbon compound. Now the second component is a nitrogen base. Nitrogen bases are of two categories, purines and pyrimidines. Purines are double ring structures. They have a benzene ring attached to this, an imidazole ring is there. So two rings are present in their skeleton. And purine nitrogen bases are adenine and guanine. guanine are purine bases. And the pyrimidine bases are single ring structures. They are thymine and cytosine. So adenine and guanine are purines and thymine and cytosine are pyrimidines. These are the four nitrogen bases coming under two categories. Okay. A nitrogen base can combine with a sugar. Nitrogen base, when combines with a deoxyribose sugar, it becomes a nucleoside. It becomes a nucleoside. A nitrogen base combines with the sugar at the first carbon atom. At the first carbon atom of the deoxyribose sugar with a chemical bond called N-glycosidic bond. The name of the bond is N-glycosidic bond. And then it becomes a nucleoside. So sugar plus nitrogen base is a nucleoside. And based on the type of nitrogen base combines with the deoxyribose sugar, there are four different nucleosides. If adenine is the nitrogen base, combining with the sugar molecule, then the resultant nucleoside is adenosine. If it is guanine, the nucleoside is guanosine. And if the nitrogen base is thymine, the nucleoside is thymidine. And if the nitrogen base is cytosine, the nucleoside is cytidine. So in that way, there would be four nucleosides in DNA molecule. Okay. Now the next component is the phosphate group. Phosphate group. A phosphate group is PO4. It's a radical PO4. Can attach with the sugar molecule at 2 points. Phosphate group can attach with the sugar molecule at the third carbon atom and also at the fifth carbon atom. So the third carbon atom, sugar molecule can combine with the phosphate group, and at the fifth carbon atom also, a sugar molecule can combine with a phosphate group. So a sugar molecule combined with a nitrogen base at the first carbon atom and a phosphate group at the third carbon atom or the fifth carbon atom is called a nucleotide. Then it becomes a nucleotide. The combination of all these three components results into the formation of a nucleotide. Again, based on the nature of the nitrogen base involved in the formation of the nucleotide, the name of the nucleotide varies. If adenine is the nitrogen base, then the nucleotide is adenylic acid. Adenylic acid or adenylate. If guanine is the nucleoside or guanosine is the nucleoside or guanine is the nitrogen base, then the nucleotide formed is guanylic acid. If thymine is the nitrogen base in the nucleotide, then the nucleotide is thymidinic acid. Thymidinic acid. If it is cytosine, it is cytidinic acid. So we have four nucleotides with four different nitrogen bases. So when we examine the structure of the nucleotide, we can see that sugar is common in all the sugar and phosphate are common in all the nucleotides. The varying component is the nitrogen base. So we can say in a DNA molecule, there is a sugar phosphate backbone. So sugar phosphate component acts as the backbone of the DNA molecule or backbone of the nucleotide chain. Then to form a polymer, so DNA molecule has two polymer chains connected together, and each polymer chain, each strand, we say the strand, each strand of the DNA molecule is made up of many nucleotides connected together, many nucleotides. So one nucleotide is connected with the other nucleotide, another nucleotide, by means of a bond. By means of a bond established between the phosphate group at the third carbon atom of one nucleotide and the phosphate group at the fifth carbon atom of the preceding nucleotide. That chemical bond is called a phosphoester bond. Phosphoester bond. So, a single phosphate group is connected to two nucleotides. The phosphate group at the third carbon atom of one nucleotide is also connected with the fifth carbon atom of the next or the preceding nucleotide by phosphoester bond. So, between two nucleotides, two phosphoester bonds should be there. So, that condition, that is called a phosphodiester bond. Phosphodiester bond. A phosphate group is connected to the third or fifth carbon atom of a nucleotide is by phosphoester bond. But two nucleotides are connected together by means of a phosphodiester bond, that is, a phosphoester bond between the phosphate group located on the third carbon atom of one nucleotide and another phosphoester bond established between the phosphate group and the fifth carbon atom of the preceding nucleotide. So actually, this is the phosphate group that connects the two nucleotides together by means of phosphoester bonds, two phosphoester bonds. So phosphate group is the link, connecting link between the two nucleotides, as you can see in the diagram here. So in that way, nucleotides are connected one after another to form a long chain of polynucleotide. Long chain of nucleotide, that is called, then it becomes the polynucleotide chain. Polynucleotide chain. When we examine a polynucleotide chain, we can see the polynucleotide chain or the strand is polar. That means it has polarity. It has a polarity. In the first nucleotide of the nucleotide chain, the fifth carbon atom of the sugar molecule of the first nucleotide, the fifth carbon atom of the first nucleotide, fifth carbon atom of the sugar of the first nucleotide would have a phosphate group, a free phosphate group, without being connected to any other nucleotide. So that phosphate is free. That end of the nucleotide is called a 5' end. That is called a 5' end. 5' end. And the third carbon atom of the last nucleotide, third carbon atom of the sugar of the last nucleotide would have a free OH group. It would have a free OH group without a phosphate group there. That end is called the 3' end. It's called a 3' end. So, every strand, each strand in the DNA molecule possesses two distinct ends, a 5' end and a 3' end. A 5' end and a 3' end. So we can say the strands have polarity. Strands have a polarity. The two strands in the DNA molecule are connected together by means of hydrogen bonds between the nitrogen bases. So in the DNA molecule, the two strands stand side by side, is located side by side, with the nitrogen bases facing each other. With the nitrogen bases facing each other. Between the nitrogen bases, hydrogen bonds are established. And by means of these hydrogen bonds, the nitrogen base of one strand or one nucleotide is connected with the nitrogen base of the opposite strand or opposite nucleotide. And this chemical bond or this connection between the nitrogen bases of the opposite nucleotides is called base pairing. It is called base pairing. And there is a rule for this base pairing. All nitrogen bases cannot pair with all nitrogen bases of the other strand. And the rule is the nitrogen base adenine always pairs with the thymine of the opposite strand. And the nitrogen base guanine always pairs with the cytosine of the other strand. So there is a AT pairing or adenine thymine pairing and a GC pairing or guanine cytosine pairing. Between adenine and thymine, between adenine of one strand and thymine of the opposite strand, two hydrogen bonds are present, or they are paired by two hydrogen bonds, connected by two hydrogen bonds, whereas guanine pairs with the cytosine or bonded with the cytosine by means of three hydrogen bonds. So you can see two hydrogen bonds between adenine and thymine and three hydrogen bonds between guanine and cytosine. So this is called a base pairing. So by base pairing, one strand of the DNA molecule is attached or connected to the next strand of the DNA molecule to form a ladder-like structure. A ladder-like structure. So DNA is a molecular ladder where the railings of the ladder, the side railings of the ladder are made up of sugar phosphate backbone, and the steps of the ladder are made up of base pairing between the nitrogen bases. Okay. And this ladder is not a straight one. This ladder has a right-handed coiling. It is coiled at a right-hand fashion. And after a very particular length of the DNA molecule, it turns to the right side. And again, after a particular length of the DNA molecule, again turns to the right side. So like that, it is a helical structure. It becomes a helical structure. Helical structure. And the distance between the two turn is called the pitch of the DNA molecule. The length of one pitch is 3.4 nanometer, or the distance between two turns in a DNA molecule is 3.4 nanometer. 1 nanometer is 10 to the power of minus 9 meter. That means it is a very small portion of a meter. 10 to the power of minus 9 meter. That is 1 nanometer. Okay. And there are 10 base pairings in one pitch. So between two turns of the DNA molecule, there are 10 base pairs. So distance between two base pairs. So the distance of these 10 base pairs is 3.4 nanometer. That is one pitch. So in one pitch, 10 base pairs are there. So distance between two base pairs is 3.4 nanometer by 10. That is 0.34 nanometer. That's the distance between two base pairs in a DNA molecule. And we can say the strands of the DNA molecule are complementary to each other. The strands of the DNA molecule are complementary to each other. What do you mean by that? Complementarity. Complementarity is the one feature of the DNA molecule. That means, if we know the nitrogen bases of one strand, we can guess the nitrogen bases, the sequence of nitrogen bases in the opposite strand. Why is it so? Because adenine always pairs with thymine, or thymine always pairs with adenine. The base pairing is between only adenine and thymine. The same way, base pairing is only between guanine and cytosine. If thymine is the nitrogen base in one strand, the opposite strand would have adenine as a nitrogen base. That is for sure. If guanine is the nitrogen base here in one DNA strand, the opposite strand would pair with the cytosine. So, if we know the nitrogen base sequence of one strand, we can guess the nitrogen base sequence in the opposite strand. So that is called complementarity. So that is why it is called DNA strands are complementary to each other. It is complementary. And we say the DNA strands are anti-parallel. Anti-parallel. We observe the diagram closely. We can see the orientation of the sugar molecule is different in different strands. In one strand, the oxygen atom of the sugar molecule is directed upwards, and the other strand, the oxygen of the sugar molecule is directed downwards. As a result, the position of the carbon atoms change. The first carbon atom of the sugar molecule of one strand opposes or faces the first carbon atom of the opposite strand, sugar of the opposite strand, and the polarity also reverses. The 5' end and the 3' end. The 5' end of one strand opposes the 3' end of the opposite strand. The same way, the 3' end of one strand opposes the 5' end of the opposite strand, complementary strand. So that is why it is called, the strands are anti-parallel. The DNA strands are anti-parallel. So DNA strands are complementary to each other and they are anti-parallel. Okay. Now, it was in 1869, the first glimpse of nucleic acid. DNA is a nucleic acid. DNA and RNA are called nucleic acids. The first glimpse or first indication of the presence of these nucleic acids in the nucleus was given by Friedrich Miescher. The scientist called Friedrich Miescher in the year 1869. And he simply called it nuclein. He simply called it nuclein. And investigations went on, studies went on, and in the year 1950, a scientist, Erwin Chargaff, proposed that there is a definite proportion between the number of adenine, thymine and guanine cytosine in the DNA. The number of adenine will be equal to number of thymine and the number of guanine would be equal to number of cytosine in any DNA molecule. And this is called the Chargaff's rule. Chargaff's rule. That was in 1950. And in 1951, two scientists, Maurice Wilkins and Rosalind Franklin, proposed base pairing in DNA molecule with the help of X-ray diffraction technique. So they established the fact that base pairing between adenine and thymine and base pairing between guanine and cytosine do occur or is there in the DNA molecule. They established this by using X-ray diffraction techniques. And by using these informations provided by Erwin Chargaff, Maurice Wilkins and Rosalind Franklin, in the year 1953, two scientists, James Watson and Francis Crick, proposed the double helical structure of DNA. So they proposed the double helical structure of the DNA molecule in 1953. And they were awarded Nobel Prize for their finding. Along with James Watson and Francis Crick, Maurice Wilkins also was awarded the Nobel Prize for establishing the molecular structure of DNA molecule. And after establishing the structure of the DNA molecule, Francis Crick proposed a very revolutionary proposition that was called the central dogma. And he told that DNA is the carrier of informations. Informations regarding every phenotypic expression or every features, every activities in a living body is coded, is contained in the DNA molecule. And this molecule, this information is passed on to the RNA. And RNA, by using this information, synthesizes the protein or enzymes. And enzymes act as catalysts to conduct the chemical reactions that ultimately results into a phenotypic expression or a characteristic feature in the body. So this is called the flow of information. And this flow of information is from DNA to RNA. Informations are stored in the DNA, and the stored information in the DNA in the coded form, in some coded forms are transferred to the RNA, and RNA, by using this information, synthesizes the protein. So the flow of information from DNA to RNA to protein. And this is called the central dogma. It was proposed by Francis Crick. It is called central dogma because every living organism, whatever may be the organizational level of the organism, whatever may be the biological status of the organism, this central dogma is common to every living organism, including viruses. So that is why this is called the central dogma. In short, life is, or the phenomenon of life itself is based on this flow of information or the central dogma. Okay. Now let us discuss some more features of the DNA molecule. The length of the DNA molecule, the size of the DNA molecule is determined by the number of nucleotides present in a DNA strand. The number of nucleotides present in one strand will be equal to the number of DNA nucleotides present in the next strand, in the complementary strand also. So the number of nucleotides present in a strand of the DNA molecule determines the length of the DNA molecule. Some examples are given in your textbook. The bacteriophage 5-174. One bacteriophage 5-174 has 5,386 nucleotides in its DNA molecule. 5,386 nucleotides in its DNA molecule. The bacteriophage lambda, another bacteriophage lambda, has 4,805 48,502 nucleotides. 48,502 nucleotides in bacteriophage lambda. In bacteria, in Escherichia coli bacteria, in E. coli bacteria, the number of nucleotides are 4.6 into 10 to the power of 6. It's a large number of nucleotides are present in the bacteria compared to these bacteriophages. In E. coli, 4.6 into 10 to the power of 6 number of nucleotides are present. That means the DNA is a lengthy DNA compared to the DNA of the bacteriophages 5 and lambda. In human being, the number of nucleotides are 3.3 into 10 to the power of 9 in the haploid condition. 3.3 into 10 to the power of 9 nucleotides in the haploid condition. And in the diploid condition, we have 6.6 into 10 to the power of 9 nucleotides. So the length of the size of the DNA molecule depends upon the number of nucleotides. Okay. We shall wind up the class here.